Hello, welcome to this new course named Functional Genomics. My name is Ganesh. I am a professor at the Indian Institute of Technology, Kanpur. So, I have been teaching in this institute biology, genetics, molecular biology, human molecular genetics, and my research focus has been in understanding changes in the DNA and how it could affect the way our brain functions, especially the one that relates to the disorders. So, our group has been working on neurodegenerative disorders that run in the family, dissecting the pathways and other important signaling mechanism. So, you can look into our website that is linked in this course portal to know more about us. So, coming back to this course titled Functional Genomics, which is a contemporary topic because so far you must have heard about books and courses that have the name called Genomics, which pretty much talks about the genome. So, if you look into the evolution of how our understanding of our genome has evolved from Mendel to what you understand now, what we are seeing is that in the previous years people looked into the phenotype, that is the change that you see in the organism, for example in fruit fly, or in case of humans that is a disorder, and then you try to go and map and find what is the genotype, what is the gene whose change causes the disorder. So, that was the genetics, and then people started looking into the genome, whole genome, understanding the sequence, and that is pretty much is done in 2004-5. Majority of the genome sequence has been complete. Now we sort of know the sequence, but that has really thrown a new challenge as to what the sequence mean. The genome is not static because the sequence that we have captured is a snapshot. We just got the sequence, but that's the sequence per se does not give you any information. So, it's a very dynamic organelle, dynamic in the sense it keeps sending a signal in the form of RNA. Some of the RNA transmit the signal as protein, some of them function as RNA, and some protein when it is made, they may be modified to make it more active, less active, or to degrade. So, it is a very dynamic state. So, how will you understand the function of the genome, not in the genomic context, the genomic sequence, but in terms of its function, right? So, that has been the question, and large number of studies have come in the recent years, especially last 10 years, where people have tried to develop tools to understand the dynamics of the genome in terms of what kind of changes happens, not necessarily the sequence, but how the DNA is modified, for example methylation, or how the chromatin, meaning the protein DNA complex, how that is modified in terms of acetylation or methylation or ubiquitination, and how that chromatin alterations can change the way the gene functions, or the transcripts, for example, they can undergo splicing, there could be variation in the level. So, how you can really measure these changes. And third aspect is there could be variations from tissue to tissue, cell to cell. So, these variations really can play a huge role as to how the tissue functions, how the organ system functions or organism functions, and many other such applications. So, that would be the focus of the course, and if this is a 10-hour course, we are going to have lectures, and we will give you a lot of reading material, and we will have assignments, and of course, we are going to have exams. But in addition, we will have hangout sessions to answer some of your questions. We will have it every week, details of which will be available in the portal. And what you may get out of this course at the end of the 4 weeks is to understand how the field has changed from genetics to genomics to what you call as a functional genomics, how it has changed, how various methods, including for example computational tools, have come in to help us to understand our genome. So, this would give you an overview of the emerging topic, which will help you to do well in your exams if you are doing any of such course, to take up career in research, and of course in DNA diagnostics and other such biopharma applications if you have companies you would like to go. So, these concepts are going to really help you in performing the experiments or performing or giving a better performance in the interview and so on. I am sure this is going to be of much help to you because this is an evolving and contemporary field there are not many textbook available, so we would rely more on the online material that are available, mainly the review papers and so on. To help me in running the course, we have two of my senior PhD students, rather they have completed their PhD, who would help me in running the course. One is Anshika Goenka, and we have Anupama Rai, and they are there to help you with any of the queries. They will be there in the Hangout session to answer your questions and to for the overall maintaining the portal. So they would be there. So please do not hesitate to contact any one of us in case if you have any query with regard to the course structure and any concept if it is not clear. And I am sure you are going to have a nice time, and I would request my students to introduce themselves and say a few words.
Hi, I am Anshika. I'll be myself and Anupama will be your teaching assistants for this functional genomics course, and we'll be helping you majorly through the assignments, which will help you prepare well for the exams. And we'll also be interacting you on the Hangout sessions, which will be conducted weekly. So if you have any problems with the course, you can write to us directly, and we'll surely answer your queries as soon as possible. So, Anupama?
Yeah, the same. So we'll be there for you always, and in case of any query, you feel free to write us.
Thank you.